Good evening, ladies and gentlemen. Welcome to the auspicious occasion of the inaugural lecture of Professor Hans Strijdom. I will now invite you to stand as we observe the entrance of the academic procession.
Please be seated. Can you hear me? Yes, sorry. Okay, while the last remaining people come in, good evening, colleagues, ladies and gentlemen. It's a pleasure for me to welcome you this evening. For those of you who don't know me, my name is Nico van Pittius. I'm the Vice Dean for Research and Internationalization of the Faculty of Medicine and Health Sciences of Stellenbosch University. It's my distinct pleasure on behalf of Stellenbosch University to welcome you all here this evening on this auspicious occasion, the professorial inaugural lecture of Professor Hans Strijdom. A special word of welcome to our Deputy Vice Chancellor of Research, Innovation and Postgraduate Studies, Professor Sibusiso Moyo, our Dean, Professor Elmi Miller, other members of the Dean's Management Team, and our Emeritus Dean, Professor Jimmy Walmink. And of course, our inaugural lecturer this evening, Professor Hans Strijdom. Dankie Hans. Welkom ook aan alle familie, vriende en collega's van Professor Strijdom, wat vanaand in persoon en ook aanlyn teenwoordig is, en insluitend Professors André Olofse en Marina de Kok van IWK, Professor Sandrine Lecoeur from UCT, Dr. Dirk Bester van CPUT, Dr. Johan Louw and Professor Christo Miller, Rabia Johnson and Carmen Pfeiffer from the South African Medical Research Council and other colleagues from the MRC, Professor Willie Daniels from WITS, en al sy ander familie en vriende, collega's en studente. We also welcome our colleagues from Stellenbosch campus and all other guests from outside the university. Today we gather here to celebrate not only the remarkable achievements that propelled Professor Strijdom to his well-deserved elevation into the full professorship, but also to honor the essence of intellectual curiosity and academic excellence that characterizes both him and our esteemed institution. An inaugural lecture marks the momentous juncture in an academic's voyage as a scholar, a researcher, an educator. It stands as a celebration of the ascent to the rank of full professorship, providing the individual not only with an opportunity to present their academic pursuits, their interests, their research, passions, and their contributions to the field of study, but also an opportunity to provide the audience a glimpse into the fabric of their character and the path that they've traversed to attain this remarkable distinction. Thus, this occasion transcends being solely a celebration of Professor Strijdam's achievements. It also serves as an opportunity to acquaint his colleagues, his students, his family, his friends, and the broader academic community with him and his work in a personal and devoted manner. Inaugural lectures are a central part of our university's academic tapestry. They epitomize a pivotal milestone in any academic's journey, and as such, this event is really a special occasion for us. But not only for us, for Professor Strijdom and for his family and friends, everyone here this evening. So we're very happy and glad that you have chosen to be part of this event and to join us this evening, sharing in the significance of this occasion with us and Professor Strijdom. I invite you to enjoy the evening. Please sit back, engage, reflect, ponder, be inspired by the insights that Professor Strijdom will share with us this evening, and I would like to now hand over to Professor Elmi Miller, our Dean, to please introduce the candidate. Thank you.
Ladies and gentlemen, distinguished guests, esteemed colleagues, with an overwhelming sense of excitement and anticipation, we have convened today to introduce an exceptional individual whose academic journey embodies a remarkable evolution from clinician to academic luminary. It's my distinct privilege to introduce to you Professor Hans Strijdom, a formidable figure whose voyage has propelled him to the esteemed position of Professor and Head of the Division of Medical Physiology within Stellenbosch University's Faculty of Medicine and Health Sciences. Furthermore, he assumes the pivotal role of Deputy Director at the Center for Cardiometabolic Research in Africa, a testament to his multifaceted leadership. Professor Strijdam's academic expedition mirrors the intricate vasculature he so passionately studies, an odyssey marked by twists of fate, moments of revelation, and the turbulence of intellectual pursuit. His scholarly explorations orbit the cardiovascular realm with a steadfast focus on vascular and endothelial biology. What originated as a laboratory-based inquiry have over time metamorphosed into a vibrant body of clinical and impactful research. Noteworthy is the fact that Professor Strijdam's entire academic odyssey unfolded at Stellenbosch University, spanning from his inaugural year as a tenacious MB-CHB student in 1987 to this present moment. His fervor for scientific inquiry ignited during his application for a B-Medsci program during his third year of MB-CHB. Among a select group of 4, he ventured into physiology and pharmacology, a memorable juncture that ignited the beacon of his academic career. Having successfully traversed the MB-CHB milestone, he embarked on an internship in KwaZulu-Natal, followed by a 2-year work experience in the UK. Upon his return to Karl Bremer Hospital, the crossroads of his destiny beckoned, offering him a lecturer position within the physiology domain. It was during this juncture that he unearthed a profound predilection for academia over clinical practice, with his research retaining a steadfast clinical underpinning. In 2015, he spearheaded the Indo-Africa Study Consortium, a collaborative effort spanning international borders, delving into the nuanced alterations within vascular endothelial systems among patients with HIV. Professor Strijdam's unwavering commitment to untangle the intricate interplay between HIV and metabolic dynamics has yielded remarkable revelations. His extensive repertoire of scholarly papers and captivating presentations stand as a testament to his important influence on the scientific narrative. It's only fitting that his endeavors have garnered the recognition they deserve, manifesting in prestigious grants from the United States NIH and the South African National Research Foundation. His lineage carries a profound political heritage, with Hans being the descendant of J.G. Strijdom, Johannes Gerhardus Strijdom, a prominent South African statesman who served as Prime Minister from 1954 to 1958. Professor Strijdam's choice of transportation, a well-known enduring BMW, resulted in student feedback motivating the faculty to pay him a bigger salary. His commitment to volunteerism extends to chairing the International Board of Youth for Understanding, an esteemed student exchange program spanning diverse nations. His journey as a school exchange student in Germany motivated him for this ongoing commitment as a volunteer for the same program. A fervent advocate of learning and teaching, Professor Strijdam's heart finds itself caught between the realms of academia and research, each occupying a cherished space. His genuine affection for both undergraduate and postgraduate students underscores this dedication. Today, we wholeheartedly extend a warm and sincere welcome to Professor Hans Strijdom as he graces the stage, ready to share his insights, experiences, and contributions. Beyond enriching the academic landscape, his work has propelled our collective comprehension of the intricate mosaic that constitutes human health, a mosaic shaped by dedication, innovation, and a quest for knowledge. Welcome, Hans.
So, Professor Müller, thank you very much for those wonderful words, and also, Nico, to you. Madam DVC for Research, Professor Moyo, Dean, Professor Müller, Emeritus Dean, Professor Volmink, and Vice Dean, for Research, Professor Nico Guy van Pitjes. Also to my colleagues from the other institutions in the neighboring parts of Cape Town. Also people coming in from Johannesburg, from WITS to join us, my former boss, Professor Daniels. Colleagues, of course, and students, and I asked both undergrad and postgraduate students to come. So I'm very glad that we are bringing the mean age of this auditorium slightly down with you guys being here. And then, of course, everybody online, friends and family. So thank you very much for this opportunity. I'm going to indeed talk to you a little bit about my journey, my academic journey, which Almi already mentioned is I built on the analogy of the vascular system. And hopefully I'll convince you that we can indeed use the body's vascular tree as an analogy for a typical academic career. First of all, this talk is dedicated to my parents. My father passed away when I was still very young, but my mother is still with us. Okay. So, what do you do when you are copied on an email from HR to your line manager telling you about, in Afrikaans we call it langdiensttoekenning, in English that's long service award. My first reaction was this. Because that entails 25 years, and it does then cause one to reflect, which I then subsequently did on the 12th of May. So yes, the mathematics from 1998 to 2023 is pretty easy to work out, but as Almi said, my path with Stellenbosch University actually started already in 1987. Now that is 36 years. And if you do a quick percentage calculation, that means that I've spent 66.6% of my lifetime in this institution. And what a wonderful 36 years it has been. And as you can imagine, full of changes, full of ups and downs, but overall, absolutely marvelous time. So the next question one should ask yourself is were these 36 years a straight line, like shown on the slide? And the answer is absolutely no. It probably more resembles something like this. And if you then carefully look, you can actually superimpose it on the vascular bed of the retina. Hence, first step one in my analogy with the vascular system. So this is more or less what a typical academic, and I'm definitely not the only one who will be able to vouch for this. So let me quickly take you through this process of 36 years. So it started in 1987, and I don't think anybody here knows that I was actually enrolled for BSc Agriculture. Not to become Farmer Hans. It was rather a situation of me having a very strong but very transient interest in genetics. And I was told, well, agriculture is the way to go, animal genetics, plant genetics, etc. But I promise you, I didn't last 2 weeks. And then I asked for a transfer, or we requested for a transfer to medicine. I don't think that would be that easy these days, but in those days, I was lucky enough to be transferred into the NBCHB program. That was a nice year in a way, but it was also filled with catastrophes as I realized that my physics, statistics, and maths marks almost made me not pass that year. The trauma was so big that I then, in 1988, went on a gap year. Now, the term gap year was not even invented in 1988. But I asked the university for special leave, one can probably call it these days. I went to Europe with a mullet hairstyle, mind you, and a moustache. Now, for the younger generation amongst us, if you guys don't know what the mullet hairstyle looks like and a moustache, it looks something like that. So to get back to business, after that, I left something out. I was also haunted by the South African Army. So I obviously did not tell the Defence Force that I was leaving the country. In those years, conscription was still at the order of the day. And they somehow managed to find me in France. How, I don't know. I got delivered a telegram from the South African Army in my domicile in France, where I was looking after a 3-month-old baby, that I need to come back and I need to go, I think it was Oudtshoorn or somewhere, and I just decided that is absolutely not going to happen. I can't remember the details of how I wiggled myself out of that one, but I remained in Europe for the rest of that year, and I returned in, don't worry about the mullet again. I returned in 1989 to resume my MBCHB degree. This was now my second MBCHB class. Only in 1991, to leave that class again and do the Bachelor of Medical Sciences in Physiology and Pharmacology, and that is where the research bug bit properly. I resumed for the third time in 1992 with my MBCHB studies, and this was going to be my last class. I wasn't going to join any new MBCHB classes. By this time I was a veteran already. I graduated in 1994, which was also a very important year for many other reasons, as we South Africans will know. So I think it was a good year to graduate. I did my internship in KwaZulu-Natal in a wonderful hospital called Ngwelezana, on the outskirts of Empangeni. And then in 96 and to 98, I did medical officer duties in the United Kingdom, and also for a short stint at the Karl Bremer Hospital before Professor Kuslach, who was our head at that stage, approached me and said, there's a position available for lecturer, do I want to apply, which I then subsequently did. There's a fast forward now, because it was a relatively uneventful path until 2006 when I graduated with my PhD. And 2020 was also a remarkable year for 3 reasons. I was then promoted to full professor. I became the head of department or head of division of medical physiology, and Prof. Fadil, Aesop, and I at last got our dream. I don't know where Fadil is sitting even. There we are. We got our dream realized with the establishment of karma. And for those of you that don't know what karma stands for, it's the Center for Cardiometabolic Research in Africa. But we can also use the other meaning of karma, as long as it's the good karma in this instance. Right, so moving on, the vascular tree, I want to just a little bit further embroider on this. So if you look at this slide, this is typical, you can find it in any textbook, but it basically gives us a nice impression of the interconnectivity of the vascular system. You've got at the center of all of this, you've got the heart, which provides the force and the energy for the blood. The blood then carries oxygen, nutrients to enrich the body. And then you can also see areas that are called trunks here of smaller blood vessels where the resistance is relatively higher. So in my view then, the analogy with a career is that we are all dependent on interconnectiveness and interdependence, like the vascular system. We also disseminate life-enriching knowledge, like the blood disseminates oxygen and nutrients. We also every now and then have to tap from our source of energy, which is in this case not the heart, but our community of colleagues and students. And finally, yes, there are going to be areas and times of resistance like we find in the smaller blood vessels of the body. Furthermore, the vascular system is also containing several different types of blood vessels, ranging from the from the very small and sometimes vulnerable capillaries, which are single-layered, to the arterioles and the more forceful and stronger muscular arteries, with the veins a little bit sitting on the side and watching the show as it goes on. The analogy there is this can represent the stages of our careers, starting with a young, naive, and vulnerable like the capillaries, into a more experienced, resilient, wise, and then often with a touch of cynicism, I suppose, older academic, which I probably have to count myself as now. Sometimes we, like the muscular arteries, we need to apply a bit of force, a little bit of initiative and leadership. And sometimes, like the veins, we just have to go with the flow and not really work very hard to get the blood back to the heart. The endothelial layer, and we're going to talk a little bit more about that later, which is the inner lining, as you can see on this slide showing now, is a protective layer. It's a single cell layer that protects basically the rest of the body from whatever the blood is carried. And believe me, the blood is not that clean as we think it is. And yes, we as academics in our careers also need to protect and defend, but we can sometimes burn out and become dysfunctional, like the endothelial cells when they get battered too much. The path can also be tortuous, like this poor person's retinal image. And then the last concept I want to introduce is the manner in which the blood flows in our blood vessels. So the optimal physiological flow is called laminar flow, as you can see. And then every now and then there's obviously also a little bit of turbulence. And once again, the road that we walk is sometimes tortuous, unpredictable, and turbulent, but hopefully always turns back to laminarity. So hopefully I have now convinced you all that the academic career is very much like our body's vascular system. So the approach to this lecture, the focus will be on my research journey, but please remember, I cannot include everything, and nor can I remember everything. I will attempt as far as possible to steer clear from too many technicalities, but sometimes it's unavoidable. And also, I think we all realize that, but research only makes up approximately 25 to 30% of my duties, of our duties as academics. And I will not try to focus on the other 70 to 75%. So I think that the context and perspective is just always important. So one can also call this talk, the rest of this talk, From Cell to Human. So for the first part, I'm going to talk a little bit about vascular biology, endothelial cells, and what I call the wonder molecule, nitric oxide. So you can see here a histological microphotograph of a rat aorta on the left. And if you look carefully, you may just be able to see the inner lining called the tunica intima, very, very appropriately, which contains this single layer of endothelial cells. And the diagrams on the right basically just shows that in a more, in better perspective. You've got the outer layer, adventitia, the media, which contains the smooth muscle cells in those vessels that have a muscle layer, and then, as I mentioned, the intima. So a few facts and figures. There are approximately 1 to 6 times 10 to the power of 13 endothelial cells in the body. Now, I Googled 10 to the power of 13th, and the outcome was mixed. Some refer to it as a trillion, and others refer to it as a word that I, for the moment, can't remember. But it's gazillion, so we call it a gazillion. Right. The point is there's a lot of them, and they make up 1 kilogram of our total body weight. If we were to strip them from all the blood vessels, they can cover 16 to 28 tennis courts. And it is the body's single largest selectively permeable biological membrane and barrier between circulating blood and extravascular cells, tissues, and organs. And as such, it is the first line of defense against harmful circulatory substances that batter it non-stop, depending on, obviously, our lifestyle. So let's talk a little bit about endothelial cells and this wonder molecule that we call nitric oxide. So I'm going to just for a moment focus on the vascular wall. I'm not going to go into details of the slide, so please just ignore all the pathways that are shown here. The message I want to bring across is that you can see at the top is the blood vessel with the blood flowing. For endothelial cells to perform optimally and function physiologically, they do require laminar flow. And that causes what we call shear stress, which is basically a frictional force that rubs against the endothelial cells as the blood flows through the blood vessels. And that serves as the main physiological stimulus for endothelial cells to release nitric oxide. And nitric oxide can then diffuse into the smooth muscle cells and allow them to relax and dilate. And this is a good thing, believe me. So we want nitric oxide to be produced by healthy endothelial cells in blood vessels that conduct blood at laminar flow. A little bit more about nitric oxide. So before the 1980s, nitric oxide was regarded as a common atmospheric pollutant found in cigarette smoking and exhaust fumes. It is a very simple diatonic gas, but in the 1980s, these three gentlemen, and they got their Nobel Prize only 10 years later, as it usually goes, but for their discoveries concerning nitric oxide as a signaling molecule in the cardiovascular system. So this was quite sensational at the time. And there was a lot of, I wouldn't say resistance, but there was a lot of debate about their findings as to how an air pollutant can actually be a biological signaling molecule. But that proved to be the case. And in fact, this is just part of the text of the Nobel Award that they received, but in red, it represented at the time an entirely new principle for signaling in biological systems. And it was also the first time that a gas was shown to serve as a signaling molecule. So basically, a healthy endothelium equals healthy cardiovascular system. So endothelial cells release nitric oxide, as we said earlier, causing relaxation of our blood vessels, but it also prevents blood clotting. It has very strong anti-inflammatory properties as well as antioxidant properties. We move now to the heart, where nitric oxide was shown to protect the heart against ischemic injury, amongst others in our very own laboratory. And Professor Lochner, who is gracing us with her presence today, was the first author of this very eloquent paper, and Professor Marais is also here, that showed that nitric oxide, and don't worry about the technical term ischemic preconditioning, what this basically says is that it protects the heart against a lack of oxygen. And this was an important finding. Problem, however, was we didn't know where this nitric oxide came from. Was it from the endothelial cells, either outside or within the heart? Was it perhaps even the muscle cells of the heart? And hence, the question, what is the source of nitric oxide in the heart? And the animation is a bit wrong here, but this is how my love-hate relationship with nitric oxide began, and so too my PhD journey. Because I then took that work a little bit further, and Professor Lochner thought it good to send me straight into the perfusion lab, which I must say, Amanda, was not my favorite place. But just to show you, what we then did was we removed these hearts from the rats. We put it on a perfusion rig, and we perfused it with an enzyme called collagenase, which basically dissolved or digests the heart into pulp, like you can see there. So in this test tube here, this is basically that heart just digested, and this is full of ventricular myocytes or cardiac muscle cells. And this is what they look like. If they're nice and healthy, I had to be a bit selective in choosing these pictures because they don't always look that nice. And then they look like this, and we call them rods, as you can see, typical of muscle cells. So what we did was we induced hypoxia based on a very simple method, which was described already in 1995. You basically just take a small little portion of that pulp, you pellet it, in other words, you centrifuge it, and you layer it with mineral oil, as easy as that. And you put it back in the incubator and for whatever period of time, and in that you induce hypoxia. And this resulted in my first PhD article in 2004, where we could show in these heart cells, muscle cells, that the enzyme producing nitric oxide did not contribute to the protection. The problem was we couldn't yet measure nitric oxide because it's a gas. So we could only look at indirect markers of nitric oxide, and in this case we also looked at the enzyme. So we excluded the myocytes as the source of the protective nitric oxide. Now I say this is a love-hate relationship, and I'm going to explain to you why we hate nitric oxide at the same time. The problem is it's a gas. It has an extremely short half-life. It can move anywhere it wants, and it is a free radical. So you can imagine how difficult it is to actually measure and detect. It's a little bit like trying to catch a chicken, okay, because it's running away from you all the time. We then set on finding a technique where we could actually directly measure intracellular nitric oxide. And I'm showing this photo. This was this faculty's very first flow cytometry. It actually probably should be in the museum right now. And Professor Miller, are you around? I saw you somewhere. Professor Miller, you will remember our afternoons and evenings that we tried to sort out this machine. But we managed, very much with the help of Christo. The reason why I'm showing this is because this tool gave us an opportunity to start measuring the intracellular nitric oxide. So it's a very cool setup. You put the cells in this test tube, it sucks it into the flow cytometer, and after weeks and weeks of trying to get the scatter plot right, and Christo played an instrumental role in that, we could eventually find a scatter plot. Now, again, don't worry about the technical details, but this red area here are what we refer to as the healthy and viable cardiomyocytes. We then treated these cells with a fluorescent probe that was specific for nitric oxide. And again, don't worry about the graphs. I have to provide evidence that what I'm saying is true, so that's why I'm putting it here. Also, I have to say, and I told Almi earlier, that it took me very long to delve into my archives to find all of these. We're talking, 10, 15 years ago. So I have to show it. But what we're trying to show here just is that as you add the cells and put them in an hypoxic environment, there's a right shift in the fluorescence, meaning that the nitric oxide production increased. And when we inhibited the enzyme that produces nitric oxide, there's again a left shift. And this then resulted in the second PhD article, which was actually quite a good one in the sense that a lot of other people used the technique like us. So this was also published in 2004. So that's the myocytes, but remember we want to get back to endothelial cells. And the reason for that is because in the heart, the myocytes and the endothelial cells of the heart are very closely located to each other. Never a distance that exceeds 2 to 3 micrometers. And there are 3 cardiac endothelial cells for every cardiac muscle cell. So as you can see in the text here at the bottom, this creates the perfect conditions for crosstalk between these cell types. And particularly nitric oxide could be a good candidate because it's a gas and it can so easily flow from one cell to the other. So this was then the next mission and the final mission of my PhD was to see if we can figure out what the relative contributions are of nitric oxide production between these cell types. And again, we used the old fax caliber in the faculty. And again, don't worry too much. We've got the fluorescence down here again. But the point I'm trying to make here is that we found that the endothelial cells produced around 26-fold to 52-fold more nitric oxide than cardiomyocytes, which they told us that they, and this is some enzyme Western blot studies that we also did just to support our findings. And this then also led to a publication where we could show that the cardiac endothelial cells are indeed a greater source of nitric oxide in the heart, also under hypoxic conditions. And this led us then to our so-called spillover hypothesis. I never got to prove this hypothesis because I wanted to move on with my research life, but I thought this would be a nice slide to end my PhD story on. There was some sharing expertise with the sperm researchers in our laboratory. They also started using that technology, and some of my colleagues and I looked further in intracellular pathways which I'm not going to focus on right now. So this took me to the next chapter in looking at endothelial dysfunction. Now, we already in the 2010 started the conversation. We put out, Amanda Genis and I, my PhD student at the time, we put out a commentary on the International Atherosclerosis Society website just to start testing the waters with regards to where the research world stands on endothelial dysfunction. We also have published an editorial and a review article now to my students. Please take note that Mashudu, the first author of this article, was my master's student at the time. She unfortunately can't join us, but hopefully she's online. But this article has so far elicited 686 citations. And I have to thank Mashudu for that. I mean, of course, I also take a little bit of the acknowledgement. But Mashudu, well done if you are here or online. That still is by far our most cited article ever written. So endothelial dysfunction can also be regarded as the early precursor of cardiovascular disease. And the reason for that is that our endothelial cells are permanently exposed to very harmful stimuli in our blood circulation, such as free radicals, LDL, which is the bad cholesterol, the breakdown products of fats, inflammatory cytokines, such as tumor necrosis factor alpha. And those of you that can think back to the dark days of COVID-19 will remember this called cytokine storm, which was often held as one of the reasons for the high mortality that we saw in those patients in the early days. And TNF-alpha was one of the culprits. High blood sugar, infective agents such as HIV, and again here SARS-CoV-2, which itself could also infect endothelial cells, nicotine, and then also some antiretroviral drugs that we use for people with HIV. Thankfully, those are less and less being used with a newer generation coming to the fore. So all of this then leads to a reduced nitric oxide production by these cells. The barrier function becomes broken. Our arteries become stiff. We get dysregulated blood clotting, increased inflammation, and also increased oxidative stress. From a clinical point of view, and that's what I really liked about going into this line, was it's early and it is potentially reversible. So if you can catch it early in a patient, you can actually reverse the dysfunction and prevent future cardiovascular disease, which makes it a relevant topic to look at further. So again, this is a very busy slide. I'm just trying to show you how the typical cardiovascular risk factors and lifestyle factors like diabetes, dyslipidemia, obesity, smoking, and hypertension can lead to endothelial dysfunction. And if we go beyond the point of return, it can spill over into cardiovascular disease itself, including hypertension, stroke, heart failure, etc. But I want to add to this list HIV and with and without antiretroviral therapy, because we're going to speak about that a bit later, as a possible risk factor for endothelial dysfunction. And then much more recently, as I said earlier, SARS-CoV-2. And in fact, just last month, a group of us published this paper in Frontiers in Immunology, showing, amongst others, that vascular endothelial dysfunction markers were associated with mortality in these patients that was seen in Tygerberg. So very relevant questions that we are asking here. So now we wanted to simulate endothelial dysfunction in the Petri dish. So I had to go back to cells again for a brief moment. And we decided to expose these cells to tumor necrosis factor alpha, which is this cytokine that I spoke about that we find in HIV and also in COVID-19. So we exposed the endothelial cells to this cytokine, and we found indeed that the nitric oxide production was reduced. We showed increased oxidative stress in those cells, increased cell death, and then I'm showing this for the nice pictures also. But here we also specifically looked at mitochondrial free radicals or reactive oxygen species. So you can see here the endothelial cells, the blue stained nuclei, and these red stains are the mitochondria containing the free radicals. So we could also show that the mitochondrial ROS production increased in these endothelial cells when exposed to TNF-alpha. We also ventured for a brief moment, Amanda, I don't know if you remember those days, into proteomics. We did find a few nice results, but I must say and we have some prominent statisticians here with us tonight, but the statistics in this became a bit too much for me at the time. Professor Gerard, unfortunately you were not, unfortunately you were not here yet. Otherwise we would have come to you. But we thought we could show that there was increased pathways linked to oxidative stress, pro-inflammatory pathways, and also pro-clotting pathways. So we were happy that we found an in vitro model that could show endothelial dysfunction. And Amanda then put all of this together in a beautiful and eloquent book chapter where we brought all of these data, because it spanned literally 4 or 5 years of data together in this book. We could also reverse, and that's the point I was trying to make earlier, that the relevant thing about endothelial dysfunction is that it is reversible. And we wanted to show it in the Petri dish as well. So we could. We treated these cells with a cholesterol drug, actually, fenofibrate, which is used for resistant dyslipidemia patients. And we could show, as you can see here clearly, that when we added fenofibrate, it returned the nitric oxide to control levels. We could reduce the mitochondrial oxidative stress, and we could also protect the cells against cell death. And Corley, who is also here tonight, put this all together in once again, a very eloquent paper and published this. So at this point, we moved from cells to blood vessels, I started a small section of our laboratory to do what we call isometric tension studies. So it's basically an organ bath. As you can see here, this is a rat aorta that was dissected in this dish here. We chop it up into little segments and we place it in the mounting hooks of the organ bath, and it literally can measure force, pressure. So we can then stimulate these aortic rings to contract, and we did that with an alpha-1 agonist, phenylephrine, which we find in our common nasal sprays, to make these little segments contract, and you can see the force then increases. And then after that, we treat it with acetylcholine, which is a neurotransmitter, as we all should know, but it also is a potent stimulator of nitric oxide release in endothelial cells, hence leading to relaxation. And this is for those of us that, what shall I say, or I have to include myself now, are about 50. Okay? So what we did in this preliminary data, this is looking at relaxation. So the 0 is at the top and 100% relaxation at the bottom. If you look at the red curve, you can see these were the old rats. They didn't relax very well. So age, and I'm not being ageist if I say this, it's a physiological phenomenon. These old rats, and yes, 32 weeks is very, very old for a rat, they could hardly relax. So their endothelial system was basically dysfunctional. Compared to the young rats, students at the back there, 100% relaxation, so obviously your endothelial cells and nitric oxide is still working pretty fine. So this was to test the system. We then went over to look at nicotine. So the rats were given fed, or injected, I think, injected nicotine, and at the same time they were fed with rooibos in their drink water or melatonin. Now, melatonin is the hormone of darkness, by the way. It's the hormone that is released by the pineal gland. But Professor Lochner, again, was one of the stalwarts in showing that melatonin is much more than just a hormone regulating our day-night cycle. It is probably one of the strongest antioxidants known to us. So we started doing a lot of work with melatonin, and what we're looking at here is the contraction of these aortic rings. And you can see when those rats that were injected with nicotine, their aortas contracted even beyond 100%. So they were hypercontractile. And look at what melatonin did at the bottom here. It almost, well, I won't say reversed because you need contraction, but it brought down considerably the contraction in these aortic rings. And then we also have the two rooibos, fermented and unfermented, that also also significantly reduce the contraction. So we could show here that rooibos and melatonin could reduce the hypercontractility induced by nicotine. And on the relaxation side, we show similar results. Now we're looking at how the blood vessel is relaxing with acetylcholine. Again, the blue line, that's nicotine. Relaxation is around 60% only, whereas the nicotine and melatonin treatment group and those with the fermented rooibos almost relaxed up to a point of 90%. And this beautiful work was also written up by a PhD student at the time, Michelle. We wrote this together with Chantal Windvogel, can't be here tonight, unfortunately, to show that we could reverse the injury caused by nicotine. And so the Africa era started. Now the Indo-Africa era, but it was the Era Africa call, was published in 2013, and this formed part of the European Union Framework Programme 7. So that was a few European Union calls before the current horizon. And they had a call. It was a very complicated call, unfortunately, so we really had to think long and hard about it. But there was a program that they called interfacing challenges. So they wanted us in our proposal to include 2 or more societal challenges that converge at the same point. Now, many people, when they come to that point, will already give up on even going for this call. But anyway, we continued. It had to have at least 2 African partners and at least 2 EU partners, but it must be led by an African partner institution. So we pushed through. We were lucky. And out of the 106 proposals at the time, 17 were selected, and the Indo-Africa that we spearheaded was one of them. And this was really a paradigm shift in my personal career, because it did bring me into touch with international research and being part of research consortia, leading research consortia. So I'm glad that you also mentioned that highlight, Almi, because it was an important milestone for me. So to get back to the core, so how did we now decide what challenges, societal challenges, are we going to use? And we decided we're going to make use of health challenges on the one side, focusing on HIV infection, and environmental challenges. And there might be many of you that don't know that we even did a lot of air pollution work. France, who is online, is not in Cape Town anymore, did the absolutely beautiful work on air pollution. I will unfortunately not have time to go through that here. So these were the 2 societal challenges, and we basically wanted to see if there's an overlap with endothelial dysfunction in humans. So why HIV-associated cardiovascular disease? Why did we choose HIV? So a couple of years ago, there was a systematic review and meta-analysis that looked at the global burden of cardiovascular disease in people living with HIV. And their conclusion was that people living with HIV are twice as likely to develop cardiovascular disease, even compared to the general population. And with its greatest impact in sub-Saharan Africa and Asia Pacific. They went further to say, and I'm on this text block here now, that in our part of the world, South Africa, and they specifically talked about South Africa, Botswana, Swaziland, and Lesotho, they estimate that more than 15% of the total cardiovascular disease burden is attributable to HIV. So it is something that we need to take note of, and particularly in our part of the world, our public health systems are under strain, and we often find that people with infectious diseases are treated for the infection and renew the tablets and look at the viral load and the CD4 count, and they forget just to take the blood pressure in these patients to see if they don't have a cardiometabolic risk. So it is a relevant topic, I think, that we wanted to pursue. This editorial in the Circulation, which is one of the top cardiovascular journals, actually proposed that it might be time to recognize HIV infection as one of the major cardiovascular risk factors, in addition to smoking and diabetes and obesity, etc. So why are people with HIV at higher risk for cardiometabolic disease or cardiovascular disease? Despite treatment, and we must admit that the antiretroviral therapy regimes have improved tremendously in recent years, and these people start living longer, if not as long as the general population. But that then immediately also places them at higher risk for developing cardiovascular disease. So there's the aging HIV population, but there's also the problem that the antiretroviral therapy can never really totally get rid of the immune dysregulation in these patients. There's always a residual chronic immune activation, and that is by itself potentially a cardiovascular risk factor. We also find that people with HIV have a higher prevalence of modifiable risk factors such as smoking, alcohol, and lately, even obesity. And then there are the antiretroviral therapy drugs with their metabolic side effects that could also play a role in causing, as I said at the bottom here, endothelial dysfunction and then therefore also cardiovascular risk. So quickly, just a consortium, 3 countries, South Africa, Austria and Belgium. I was the PI of this consortium and the project coordinator, and I had 2 colleagues in Potchefstroom, Professor Carla Fourie, and then Professor Nkechungak at Walter Sisulu University in the Eastern Cape. And this is just to show you the nice people that we were. It was quite a big team. At the kickoff meeting, Prof. Guy van Pitjes, I distinctly see your face there. You actually opened that event for us. I don't even know if you still remember that. So that was way back in December 2014. So I'm not going to stay very long on this slide, but we created groups. In those years, people with HIV were not immediately placed on therapy yet. So we had this opportunity to also include participants without treatment and follow them up compared to people on treatment. These days that is obviously not possible, and rightly so, because people need to go on to treatment immediately. So these were the 3 groups. We had a HIV-free group, we had living with HIV not on treatment, and then also HIV positive with treatment. And these are just all the stuff we looked at. I'm not going to bore you with that. What I do want to show you are the very fancy, nice equipment that introduced quite novel research technologies in the African context. So the first one was this ultrasound-based device that could measure endothelial function clinically and non-invasively. And basically, what we do is, you can see there's a blood pressure cuff on this person's arm. And here you've got the ultrasound probe. Welcome, guys, and thank you so much for coming. They just wrote a test, so I told them they're allowed to come late. So welcome. We've got the ultrasound probe that measures the relative width of the brachial artery. Now, this blood pressure cuff is a very important part of this protocol. I'll explain that in a moment. So for the first minute or so, the patient just lies there with a probe, nothing happens. You visualize the brachial artery, as you can see there. And then for 5 minutes, you hyperinflate that cuff to induce ischemia in the arm. So for 5 minutes, there's no blood flow in the arm of that patient. It causes a bit of pins and needles, but they generally, Ingrid, survive it. I'm saying Ingrid because she's one of the ones that usually had to do this to the patients. So basically, you block the blood flow, and then after 5 minutes, you release that cuff. And what then happens is you have this resurgence of blood flow back into the brachial artery, which stimulates the endothelial cells to release nitric oxide and cause vasodilatation of the brachial artery. And it's that extent to which the brachial artery dilates that we are measuring here as an indicator of endothelial function. So we were pretty happy about this, and we started rolling it out. We were able to purchase 3 of these devices. I think the Rand-Euro exchange rate was still slightly more favorable in those years. And then we also introduced this very exciting new imaging technique, which we are still using up to today, the retinal imaging. So we literally take photographs of the retinas of our participants, and we focus on the blood vessels, the vascular network. Remember, the academic career pathway looks something like that. And the nice thing about the retinal vessels is that it provides us with a unique clinical research opportunity because it's non-invasive. You can visualize the microcirculation of the patient, and we know after many years of research that changes in the retinal microcirculation can serve as a surrogate marker of, amongst others, hypertension, diabetes, obesity, and coronary artery disease. So a nice tool. It's almost like a window into the vascular system of our body. So we included that as well. Now, showing this photo, these are my current, my master's student, Kaya, and Tahira, postdoc. They seem, as you can see, very impressed with this new gadget that they're learning to work on. But there's another reason. Tahira, sorry, I'm going to have to embarrass you now. But ladies and gents, it's Tahira's birthday today. So I think let's just give her another. Where's Tahira? Okay, so she's sitting at the back. So Tahira is leading our retinal imaging section at the moment. And Elmi, I think you were also asked to not pose, but they took at the open day, right? Yes. So we've got your retinal fingerprint somewhere, Madam Dean. We'll have to give it to you. So just quickly, ladies and gents, so this part I'm going to glance through the results because there's simply too many. But I just wanted to show you the concerning side of looking at cohorts like this is now. Bear in mind this was a relatively young cohort. The mean age is only about 40. And just look at this risk factor profile that we found. And this is regardless of whether they had or did not have HIV. The smoking rates up 60% plus. Central obesity, 40% plus, like I said, in all groups. Diastolic hypertension, quite high. We had this interesting phenomenon which we're still trying to figure out, is we had this very high prevalence of low HDL cholesterol. So the good cholesterol was low in a lot of our untreated HIV patients. And we still need to figure that one out. And then also quite concerning is we have this level of systemic inflammation. We met high sensitivity C-reactive protein here. Almost 60% of our participants had elevated CRP levels, which tells us that there must be some low-grade inflammation or infection or at least exposure, could also be air pollution, that causes this low grade of inflammation in our participants. Anyway, so I'm going to share just a few of the data with you. What we're looking at here, this is baseline, so it's once off, it's cross-sectional. We looked at the FMD, so this is endothelial function. And we got very excited because if I had to believe this, our participants with HIV and ART had excellent endothelial function. It was significantly better than both the other two control groups. But here, and this is again to our younger researchers, you always have to follow up your participants. Okay? So if you ever can do longitudinal studies, please do it, because then you will see, you could then also sometimes see this. The trends that we saw baseline over 18 months started dissipating. It was still slightly higher, but not significantly so anymore. So again, never get too excited with cross-sectional data. It's important, it can lead to something else, but one always has to try and see if you can follow them up over time. And we found something similar with the retinal venules. Now the rule of thumb with the retinal venules is the wider they are, the higher risk the patients have. And of course, the narrower they are, the less risk. And again, we found that our patients with on ART had pretty healthy retinal venules if we had to go according to this data. And for this snapshot, it was obviously true. But again, when we followed them up after 18 months, we saw that differences also started equaling out. Finally, just the importance of checking the viral loads in our patients with HIV. So what we did here, we stratified our viral loads into high, medium, and low. And you could see here that a group with a very high viral loads, in other words, they were not well controlled by their treatment, had poor endothelial function. They had wider retinal venules. And we also looked at the IMT. Now, the IMT is the intermediate thickness of the carotid artery, which is a kind of preclinical atherosclerosis, and it was the highest in our participants that were really controlled. So the association between viral load, checking the viral load, and cardiovascular risk is really important. And this is one of the take-home messages that we would like to take forward. So there were a host of publications, and I'm showing it not to brag, but just to show that each and every single of these publications were written by my students. And I think that is something that we all should strive for. All of them were first authors. Festus is France, another one by France. This was with Professor de Kloet. We had a collaboration with, also it was a medical student actually that was the first author. And again, another paper by France. And then also our colleagues at UCT that we collaborated with got this very nice review paper out. So all in all, a very successful Indo-Africa. We're still busy figuring out the 54. We're at the 54 months data now. Unfortunately, COVID threw us a curveball. We lost a lot of our patients in the follow-up because of COVID and lockdown. But we'll see what if there's any sense that we can make with that data. And then the next chapter just arrived last year, and the lady that helped me get the award, Helena, I will be eternally thankful to you for this National Institutes of Health Fogarty grant that we received. It's an exploratory study that we're going to do looking at mitochondrial function and see if that's perhaps a potential underlying mechanism in the cardiometabolic disease that these people have. And Gerald, my colleague, who's our mitochondrial expert, is a part of the team. So we're going to look in the South African cohort and also in Kenya. Fingers crossed that we're going to get interesting data and that we will be able to figure out more about the mechanisms. Right, so that's the research side of things. I hope I didn't bore you too much and that you are convinced that these are important topics to look at. I just want to make a few random remarks. And the first thing is to my students, who, like I said, I specifically invited some of our students to be here tonight. Both the undergrad and the postgrad students must know the reason why I come to work every day is because of you. Teaching and mentoring, and then sometimes educating is unfortunately also required, are just as important to me as my research. And the 36 years association with Stellenbosch has made me eternally grateful to this fantastic institution. Important people, my work mom, as I used to call her, Professor Lochner was my PhD supervisor and mentor. Professor Kuslach, may his soul rest in peace, who passed away. He was head of department for us for many years. Jimmy, you are also one of important people in my career. And Professor Waltzel, who cannot be here, unfortunately. He's traveling back from overseas. There are many others, of course, but specifically in terms of forming my academic career, thank you to all of these people. My colleagues, I don't think any other division or department can boast with such beautiful people. And if you don't agree with me, then allow me to believe that. This is our wonderful and amazing division and colleagues. Again, you are also part of the reason why I come to work every day. Don't think it's only about the students. So thank you to all of you. And then my good karma partner in crime. Now, Fadil, I apologize. The photo's a bit blurry. I had to zoom. I'm looking as if I'm enjoying something there. I'm not quite sure what it is. But it was probably just because it was nice to stand next to my partner in crime. So Fadil, also to you, thank you for the road you're walking. And then my research team, they are all here tonight, or most of them at least. The enthusiasm, the energy that you bring, and this is my current team. Of course, there have many teams before them, and I've just trying to mention a few names who I saw on the guest list online. Most of them, Mashudu, I mentioned earlier, Niku, Mashele, Corley is here. Sana Charania, who's in Johannesburg, she can't be here. Frans Everson is also couldn't be here. Cassidy, I think I saw Jody is here. Shanae, I saw Yusra, etc. So again, postgraduate students and undergraduate students. My academic life will be very boring without you. And that is the end of my story. Thank you very much for your attention, and thank you for the opportunity.
Okay, good evening, everyone. Well, but you're not excited, but I think this was the most powerful lecture I've attended. And it's not saying the others were bad, but I think, I could understand it easily. So I think I'll come to one of your classes one day. So please, let's give him a big hand and clap for the lecture. Thank you. Okay, so I was introduced earlier. So my name is Sibusisa Moyo. I'm the DVC for Research, Innovation, and Postgraduate Studies at Stellenbosch University. I'm a mathematician by background, so I must say that I wish that the people who teach applied maths could work with the medical doctors to show the math students what the actual arteries and vessels look like, because we do a lot of modeling of what's happening in terms of fluid flow, blood flow, but it was really interesting to see how you break it down and make it understandable to everybody. So I really want to take this opportunity to thank everyone and acknowledge, of course, the Dean of the Faculty of Medicine and Health Sciences, Professor Elmi Muller, and also the Vice Dean for Research, Professor Nico Hefan-Pitius. He always tries to give me lessons on that. I think I'm becoming really good at it. I always practice about it. And then, of course, Professor Gerard Vassell, who's not here today, our esteemed guests, staff, students, and friends of the university, and of course, our special guest today, Professor Hans Strijdom as well. And I want to really acknowledge our former dean, of course, Professor Jimmy Vomick, whom I've also been told that is going to be heading the Wellcome Trust, right? I have to say it loud because I just couldn't keep it to myself. I think we should say congratulations, yeah? And thank him as well. Because it does show us as a university just the talent that we have here. So I'm sorry for taking the secret out, but I sort of couldn't just keep it to myself. I'm good at keeping secrets, but not the very good parts. So, so anyway, again, I just want to say congratulations once again, Professor Hans Strijdom, for this, for your promotion to the rank of full professor, of course, in the Faculty of Medicine and Health Sciences. I saw that you got your full professorship in 2020, I'm sure during COVID. And then we had to wait 3 years almost, to hear the talk. But we really thank you for giving this lecture for us, for delivering the lecture on the vascular tree of an academic career, tortuous, turbulent, and dysfunctional. I wanted to replace dysfunctional with functional because I was hoping that somewhere it's functional at some point. Okay. And then interspersed, of course, with glorious moments of laminar flow. Today, of course, for us as a university, as has been mentioned earlier, it marks a very important day in the life of the university, in the life of the inaugurant, of course, in the life of the faculty and university at large, as we come together to celebrate the ascension of Professor Hans Strijdom to the rank of full professor at Stellenbosch University. It is a celebration of academic excellence. Excellence is one of our values, and I think as medical doctors, you know our values. And since there are students here, I think behind there, maybe if we can just remind ourselves what the values are. We use E-care, a small e and then care. E for, who remembers what it is? E for what? Excellence. Okay, we'll start with excellence. So you've used one E already. And then C for? Compassion, yes. And we've seen all of these in terms of the lecture today. And we know that for medical doctors and health practitioners, compassion is so important. And A for what? A for?
Come on, guys.
Come on. E-care. A for? Accountability. Thank you. Don't Google, right? I think you've got it. Okay. And then we've got R now. We are on R. R for? Yes. Now the last E. Excellence. Okay, you started with, what was the first E? Ethics. You've done excellence and ethics, okay? So thank you so much. I think it's important for us to remind ourselves of the values that we have as a university. And you can see these in terms of the work that our inaugurant today illustrated to us. So of course, I think the other thing to remember is that Professor Hans has published extensively. I mean, you shared some of the critical outputs for us, but it's over 53 peer-reviewed articles, presented over 80 papers at national, international conferences. I say this because we have younger researchers here just to see, the examples of what makes up an excellent scholar in our environment and in academia as well. And he has done many, conferences and has spoken as invited speaker. In fact, I want to commit to invite him as a speaker at the maths conference, which is coming up, so I'll have to negotiate that with him soon. So he has supervised several students. It was really great to see the pool of students and the diversity of students, something which we've been really trying to push as a university. And I think as part of my KPAs, I'd promised to do so many things, but I can see that if I just become his best friend, then we'll meet the KPAs. So thank you so much for the passion you have for training the next and younger generation of students. So of course, we know that the inaugural lecture is a big milestone for us as a university. And it also represents official recognition as part of the university of the achievements, outstanding achievements made. And so on behalf of the rectorate, I once again want to just say thank you and congratulate Prof. Strijdom for this achievement. There are some few terms that I took out, which I'll probably use in my as I boast around about some of the things I know. So for example, I do like the chart which shows the nave, where all these things come from. I think Professor Muller will sort of relate to some of the things in leadership, right? And I think we should do then our leadership book, but using this analogy of, of what we've been told today. And I think that is really interesting, the interconnectedness, interdependence, I think, which is important. I think also realizing that the endothelial cells and relaxation, I think if you want to have a higher flow, you probably need to be more relaxed, right? Not stressed. Yeah, I will not go for any test, I think, in my next 3 or 4 years, just in case of the results that might show thereafter. So in any case, yeah, just to say once again, thank you so much and congratulations. Now I'm going to ask you again to give a very better clap than you gave. The first time it sounded like we are distant from ourselves. So please, let's clap again and say congratulations again to Professor Vassal. Thank you so much. Let's give you congratulations. Thank you so much. Thank you.
Thank you, colleagues. Thank you, Professor Moyo. Hans, I'm not sure if you measured old rats who are retired. Maybe emeritus rats might be a bit more relaxed. Oh, I thought you stood up for me. Thank you. I'm nearly done. You can sit. Just a minute or two, sir, and then we'll stand up. Thank you. Colleagues and friends, I trust that you found this event as enriching and captivating as I did, and that it also offered you a moment of contemplation on the transformative potential of education and research. It was also a clear exhibition of the profound impact that dedicated scholars like Professor Strijdom have on our society and the world at large. So thank you, Hans, from my side also. Throughout his career, Hans has inspired countless students to think critically, creatively, igniting and nurturing their passion for science and medicine. I don't think it was mentioned this evening, but his exceptional contributions to teaching have led to his consecutive nominations over more than a decade. I'm not sure how many times. It's probably 10, 12 times, maybe even more. of Hans specifically as the lecturer who made the biggest contribution to the academic achievements of our first-year students. And so that's a remarkable achievement in itself, and a wonderful accolade. Thank you, Hans. So your comment about him being a good teacher, I think, reflects also in that, Prof. Moyo. So I myself have been privileged to have known Hans for 25 years, because I also, and I didn't realize that we started at the same time, I also started the faculty in 1998 as a research assistant under Professor Albert Bayes. And it's been a privilege to have known Hans for this long time. And we in the faculty and the university are fortunate to have him as a cherished member of our academic community and now our full professorate. So, dankie Hans, en baie hartlike dank ook aan jou familie, jou vriende, jou mentors, jou kollegas onder wie se ondersteuning ek is seker en jy het self daarvan getuig, jou prestasie sekerlik nie moontlik sou wees nie. So, baie dankie. Ladies and gentlemen, colleagues and friends, all that remains for me is to thank the organizers of this evening for their hard work. Our sincere gratitude to the whole team, especially Ms. Megan Salon and the members of the Marketing and Communications Division, the colleagues in the audiovisual assistance team, the campus security, and everyone else for their assistance, and everyone else who's contributed to making this a special evening. So with this, I declare these proceedings closed. I would like to now invite you all for some light refreshments, which is served in the Dean's reception area. Thank you very much for coming, and enjoy the rest of the evening, and please drive safely. And now you can stand. Thank you.